All right, so let's look at the clathrin molecule. Clathrin is a trimer or triskelion, and it's actually made up of both heavy chains and little bitty light chains. And the light chains sit on top of the heavy chains, and they fit together very nicely in this three-legged structure. There's quite a bit of interaction of clathrin with other molecules. The light chain reacts with oxalin and ATP and heat shock protein 70, which is part of the dissociating mechanism. And the N-terminal of each of these legs is phosphorylated and binds also calcium and calmodulin. So one can imagine there's regulation there as well. What's interesting is that this triskelion is fairly planar, but it has some flexibility in the plane, so it's not perfectly planar, and therefore when these long distal leg segments like to bind to each other, so you can see how they can interact multiple ways. You get not only hexagons, but hexagons and pentagons, because it's not perfectly planar. And when you get hexagons and pentagons, you get a very nice geometric solid, our friend the icosododecahedron, otherwise known as, you got it, a soccer ball. So it's this little bit off of planarness in the clathrin associations that gives it the ability to invaginate a spherical structure. And this last diagram just shows you the nucleating proteins. These little triangle things are the adapter proteins with the clathrin on the outside. So what about the uncoating of clathrin? If you take this clathrin triskelion with multiple associations, it takes a protein called oxalin, which basically begins to pry apart the different legs of the clathrin in an ATP dependent manner. So ATP hydrolysis basically is phosphorylating the oxalin and begins to loosen the triskelion all apart, and eventually heat shock protein 70 comes in as a chaperone to help move the proteins and change their shape enough that the individual triskelions dissociate, and then the entire coat can begin to fall off. So what about the dynamin? Pinching. How do we know about dynamin? Once it was isolated, its mechanism was investigated, again looking at its GTP dependence. We can, in isolated synaptosomal preparations, see vesicular formation, and the haze here is the clathrin coat around a vesicle. But in this particular piece, experiment, which includes cytosol to help make all of this happen, GTP gamma S, a non-hydrolyzable form of GTP, was added, so that the energy needed to actually finish the pinching off of the dynamin is not available. And you can see the dynamin ring getting larger and larger and larger and getting a long neck formed without really pinching off the vesicle. So we know that from experiments like that how the dynamin molecule works. The other important piece to think about is how we're changing the various radiuses of curvature, not only in the endocytosis but also in the exocytosis, because there's strain on the lipids for this kind of an outside curve versus an inside curve, or an outside curve versus an inside curve. And here there are changes actually in the lipids. A protein that was isolated from synaptic vesicles and presynaptic nerve terminals is endophyllin, which turns out is a lysophosphatidic acid acyltransferase. So what does that mean? It takes an arachidonyl CoA molecule, an acyl series, and it adds it onto lysophosphatidic acid, a lipid with just a single tail, so that this hydroxyl group gets replaced by the arachidonyl CoA, or the arachidonate, and the arachidonate here, and the CoA is released, you get a double tail on the phosphatidic acid, which essentially is what's changing the shape of that lipid. And by changing the shape of that lipid, you change it from an inverted triangle, or a triangle which is wide on one side, to a triangle which is wider on the other side, or a cone, inverted or not so inverted cone shape, if you want to think in 3 dimensions, to actually help the lipids attain these radiuses of curvature, which are so small in the 30 nanometer diameter vesicles. So a little bit of enzymatic help here helps as well to do the pinching. And you can see this polka dotted ring is the dynamin, which is about to be winched closed.